I'm Bonnineau from INRAE, and I will present you the result of our study and the impact of urban contamination and antibioresistance in microbial communities from aquatic ecosystems. This work has been done in collaboration between different teams from INRAE, from Limoges University, and from the Chemical Institute of Poitiers. As you may know, there is a worldwide dissemination of antibiotics and antibioresistance in the environment. In particular, the number of pharmaceuticals detected in surface water, groundwater, in even tap or drinking water is alarming. As illustrated by this map, the highest number can be found in North America and Europe and can reach up to 100-200 different molecules detected in one sample. In addition, it has also been shown that wastewater treatment plants are point source of antibiotics in surface water. In this example from Dean and colleagues, we can see the concentration in different antibiotics alongside the Seine River. The stars indicate the location of wastewater treatment plant. And we see that downstream the wastewater treatment plant, there is an increase in antibiotic concentration. Based on this previous observation from the literature, we conducted the ANTIBIOTOOLS project to investigate the fate and temporal dynamic of pharmaceutical residue in aquatic ecosystem. Our idea was to better look into the repartition of antibiotics within the different aquatic compartments in order to identify potential hotspots of bioaccumulation. We were also interested in precising the influence of wastewater treatment plants and the dynamic and repartition of pharmaceutical in the aquatic ecosystem. The presence of antibiotics in surface water as well as in sediment and biota exerts a selective pressure on microbial communities, which led to the development and dissemination of antibioresistance. This was our second question. We were wondering if the community microbial tolerance to antibiotic was correlated with the exposure. To answer this question, we selected 4 sampling sites, 2 on the Lake Geneva and 2 on the Arve River, which is a tributary of the Rhone River. On each ecosystem, we have a preserved site, here indicated in blue, and a polluted site, which was close to the wastewater treatment plant. In particular, on the Arve River, the wastewater treatment plant selected was collecting wastewater from urban area as well as from an hospital. We did 8 field campaigns over 2 years. On each field campaign, we had artificial substrata for biofilm colonization, passive sampler to collect pharmaceutical contamination from surface water, and we also collected sediment. And on this different sample, we did a series of analyses, including the determination and quantification of pharmaceutical residue, as well as a measure of antibioresistance in microbial community from biofilm and sediments. Here's the first result. We have the pharmaceutical residue in Lake Geneva, from the preserved site and from the site close to the wastewater treatment plant. And we have the concentration in the surface water on the top, in biofilm in the middle, and in sediment in the bottom. The first observation we made is that there were no differences in terms of amount of pharmaceutical in surface water and sediment between the 2 sites, which was quite unexpected. In addition, we observed higher bioaccumulation of pharmaceutical residue in biofilm from the preserved sites and from the site close to wastewater treatment plant. This concentration reached up to 1,500 nanogram per gram of biofilm, which is quite a lot. Indeed, it's more than 0.1% of contaminant bioaccumulated in biofilm. We observed temporal variation in contaminant concentration in surface water and biofilm, but it was less important for sediment. The profile of contamination were very different between the different compartments, but did not vary so much in a long time in surface water. We could identify molecules found at the highest concentration in the different compartments. It was atenolol and the antibiotic sulfaquinoxaline for surface water, while in biofilm, diclofenac and also the antibiotic sulfametazine were found at the highest concentration. In sediments, the concentrations were more evenly distributed, even though we observe a peak in diclofenac at the last sampling. If we go now to the Arve River, we have the same type of result, with the concentration pharmaceutical upstream and downstream the wastewater treatment plant. Here we observe an increase in concentration in surface water downstream the wastewater treatment plant. These differences between the two sites was not seen for the biofilm, neither for the sediments. Like for Lake Geneva, we observe a temporal variation in the level of pharmaceutical, but quite a similar profile in surface water throughout time. The molecule found at the highest concentration in the different compartments were the same as the one found in Lake Geneva. That is atenolol and sulfakinoxaline for surface water and diclofenac for biofilm. We wanted to know if there were a specific chemical signature for each of the ecosystem or for the preserved and contaminated sites. So we did a multivariate analysis on this contamination data. Here you have the result for the surface water. And as we can see, the concentration of pharmaceuticals did not allow discriminating the site based on the ecosystem type nor on the level of expected contamination. And it was the same for biofilm or sediment. This indicates that there is not a specific chemical signature linked to the site, neither the ecosystem. Our second question was to link this antibiotic contamination with antibioresistance of microbial communities. To do so, we choose to assess microbial tolerance to antibiotics through short-term toxicity assay. We collect biofilm and sediment from the field, and then in the lab did short-term toxicity tests to different type of antibiotics. We obtain a classical dose-response curve from which we focuses on the EC50, which is a concentration that led to a reduction by 50% of the biological response. And based on the peak principles, a higher EC50 indicates a higher tolerance to the contaminant tested and suggests a chronic exposure to a higher level of this contaminant. Here we have the result for the microbial tolerance in the Arve River for biofilm. Here you have the EC50 of the different antibiotic tested, and blue from the upstream community, and in yellow for the downstream community. And we can already see that there is a higher tolerance to antibiotic in biofilm community from downstream than upstream, suggesting a higher exposure to antibiotics as we observe previously. Nevertheless, this difference were not seen in the sediment. And again, this is coherent with the result of the contamination in sediment, which was relatively stable. In Lake Geneva, we couldn't see so clear differences between the preserved and impacted by wastewater treatment plant sites. Going further, we tried to link this microbial tolerance with the concentration of antibiotics in surface water. We did that with ciprofloxacin, for which we have the most data. And here you have the result, the microbial tolerance for ciprofloxacin versus the concentration of this antibiotic in surface water, with Arve River in green and Lake Geneva in blue. But here we can see there is no clear correlation between biofilm tolerance and the concentration in water. If we looked at the concentration in biofilm instead of the concentration in surface water, it seems that a slight correlation appears for the microbial community from the Arve River, but more data points are needed to confirm this first observation. Nevertheless, for Lake Geneva, the microbial tolerance is very stable, while the concentration of ciprofloxacin is highly variable. So this needs more investigation to get explanation. So the main conclusion of our studies are the following. So pharmaceutical were detected in each compartment of the of both ecosystems, and we found a different type of pharmaceutical in each compartment. We also identified that biofilm could be a potential hotspot of bioaccumulation, as illustrated by the high concentration found in Lake Geneva. Is pharmaceutical found in sediment at limited temporal variation, and the concentration were very similar between all sites and at all time. This might suggest that the observed contamination may correspond to background contamination. What about the influence of wastewater treatment plant? Unlike Geneva, it seems that there were no differences between the preserved sites and the sites close to the wastewater treatment plant. So here again, we should investigate a little bit more this case and maybe taking into account the current and the wind and how the flume of the wastewater treatment plant diffuse into the lake. In the Arve River, we clearly show that water contamination increased downstream wastewater treatment plants. The link between antibioresistance and exposure to antibiotics was not clearly shown in Lake Geneva and should be confirmed in Arve River, at least concerning ciprofloxacin. To complete this data, we would like first to add the data we had for more sampling to confirm or infirm our first observation. We also would like to better understand the different tools available to estimate antibioresistance, and to do so, we would like to compare the effective microbial tolerance as we assess by peak with the abundance of antibioresistant genes or integrons, which are more classical indicator of antibioresistance. Finally, it's important to investigate the potential impact of pharmaceutical on microbial function and biodiversity. I would like to thank all my colleagues involved in sampling and analysis, and thank you for your attention. I will be now available for questions.